So getting into section 15.3, this is stuff we haven't really covered yet, and it's about linked genes. So far we've learned basic genetic crosses. What happens when we look at dihybrid, even trihybrid, or tetrahybrid crosses, looking at how different genes will rearrange and we'll get new combinations. Remember that it's always really important to have genetic variation, a source of genetic variation somewhere, because if not, then all organisms will end up looking exactly the same, or if we don't introduce new traits, then they will be more susceptible to extinction and diseases and stuff. So it's just really important to have genetic variation. And so linked genes fall in there because they are an exception to this Mendel rule. We're going to find that some traits may be inherited together as opposed to it being completely random, which is what we've always said. So you guys probably knew there are thousands and thousands of genes, and there's just far more genes than chromosomes, right? So there's thousands of genes on each chromosome, except the Y chromosome, which doesn't have that many genes on it, just most importantly the SRY gene it has. So genes that are found on the same chromosome, which happens very often given that there's thousands of genes and only 23 chromosomes, they're called linked genes. Different than sex-linked, those are chromosomes that are found either on the X or the Y chromosome. Linked genes are just two genes that are found on the same chromosome. So our good friend Morgan, our fruit fly guy, did another experiment to examine linked genes. So he was the one who discovered sex-linked genes with the wild type red eyes with the mutant white eye fly. And so he did a second experiment to look at how how gene linkage affects the outcome. And so what he did was he crossed flies that were different in body color and wing size. So we had two different phenotypes. We had different traits with two different phenotypes. You could either have a gray body or a black body. This is the wild type, this is the mutant. Or you could have normal wings or vestigial wings, like little tiny baby shrimpy wings. So those are the two traits that we're looking at. And it talked about in the last section that the wild type is designated by a plus. So this guy has two copies of the wild type gray body allele, two bodies of the normal wing allele. This guy has two mutant alleles for black body, two mutant alleles for vestigial body. So homozygous mutant for both traits, homozygous wild type for both traits. Cross them, as you would expect, you get all dihybrids. They are heterozygous. One copy of the wild type gray body allele, one copy of the mutant black body allele, one copy of the wild type vestigial or normal wing allele, one copy of the mutant vestigial wing allele. That's what all of them look like. They're all heterozygous, all of the offspring, which is exactly what we would expect. So then he did a second test cross of the F1 wild type phenotype fly with, again, the same mutant, homozygous recessive for both mutant traits. He has black body, short wings. He crosses them again, and what you would expect to find would be these four phenotypes, right? You would expect to find doubly wild type, gray body, long wings. You would expect to find black body, vestigial wings. You would expect to find gray body, vestigial wings, and black body, normal wings, right? You should find all four phenotypes in some proportion. Because you can see that our F1 guy can make four kinds of eggs. This was a female, this was a male. So both wild type, both mutant, one mutant, one mutant. And you would get all of these right here when you cross it with our doubly mutant father. Okay, that's what we expect it to get, and it should be a relatively even ratio. Remember that it's never, or it's going to be rare if it's 250, 250, 250, 250, but we can use a chi-squared test to see if that is due to chance, okay? So the outcome that he actually got, or what we would have expected, was 1 to 1 to 1 to 1. What he actually saw was 965 to 944 to 206 to 185. So it's not 1 to 1 to 1 to 1. If we did a chi-squared analysis on this, you would see right away that this is not due to chance. Something else is going on here, which is the entire purpose of those chi-squared analysis that we did. So what we can see here is that there's far more of the parental phenotypes. One of our parents looked just like this, or had this genotype. One of our parents had this genotype. Or phenotype. One of our parents looked just like this. One of our parents looked just like this. this is a combination of the two. This is a different combination of the two. We didn't see these two phenotypes as much. So basically what Morgan was able to determine was that the parental phenotypes were inherited more often. And so these two genes, they must be on the same chromosome. So they're not separating. So on one chromosome, there's going to be the gene for wild type gray, wild type normal, and they don't get separated. So they're going to go together. Or wild type mutant, mutant black, mutant vestigial, so they go together. So we see a much closer ratio to 1-to-1 parental phenotypes, 0-to-0 recombinant phenotypes. And so these genes are not assorting independently. So this is a better picture of it. This is our maternal wild type genome. We see 2 chromosomes. We see wild type alleles on both sides. And over here we see mutant alleles on both sides. And they just don't get split up even when the chromosomes separate. So that's why we see parental genotypes that are just like, or all the offspring genotypes are just like the parents, because they're not mixing up. We're not creating new traits. This is not an example of genetic variation, okay? Our offspring are looking exactly like our parents. So that is an example of how genetic variation does not happen. But the thing is that we did still see some new recombinant organisms, right? We saw a small percentage, about, I'm not going to do the math, a small percentage of recombinant organisms, which doesn't really make sense, right? So how did we get new combinations if this is the case? And the reason for that is that genetic recombination was happening. So what this is, where is it? It doesn't say. Crossing over is happening, okay? So not very much, not enough to create normal genetic variation, but there is still crossing over. And that's why crossing over is so key in increasing genetic variation, because otherwise every single one of these flies would look just like the parent no matter what. There would be no new combinations ever. And so if for some reason both of these phenotypes are awful, and they can't survive, then the population's going out. But if for one reason you can be gray with short wings or black with normal wings and you're going to survive, then the population won't go extinct. So crossing over is super important to maintain genetic variation, especially for linked genes. So recombination is producing offspring that's different than either of the parents, has a different combination of traits. So again, we're making new combinations of traits that were not found in the parent generation. And so both of these guys, both Mendel saw this and Morgan, they relate to the chromosomal basis of recombination. So Mendel saw that in his offspring from Mendel, our good pea friend, he saw that some offspring had different combinations than their parents. So he observed this himself. And so there's the ones that look just like the parents, either in plants or flies, whichever one you were thinking about. Those are called the parental types, okay? So those offspring are the parental types. This is different than the non-parental phenotypes. So we've created a new combination of traits. These are the recombinant types, or recombinants. And you will see a 50% frequency of recombination if the two genes are on different chromosomes, because they're just assorting independently, just like you would suggest. Half the time they're going to end up together, half the time they're not. It's a completely 50-50 chance. So you can look at that in your book. It's the same thing we were just talking about. So, so while Morgan discovered that some genes can be linked, he would see that the linkage is incomplete, just like we saw. There were still some recombinant types, which were not what he was expecting. And so he was the one who proposed the process that there must be something that breaks the physical connection between the two traits, but just sometimes. And that's actually crossing over, just like we learned. So crossing over is going to happen and create recombinant chromosomes, which is how we get recombinant organisms. So let's look at this. So this parent, this is from the F1 cross. Okay, so he's heterozygous. He has 2 wild type traits, 2 mutant traits. This guy has 2 mutant traits on both of them. So as it is right now, wild type is only with wild type and mutant is only with mutant. And so if that is linked, so if there were no crossing over, we would only get mutant with mutant and wild type with wild type. There would be no combination of the two of them. And so when we cross these two together, we know that we're producing gametes, which is how we cross the two of them together. And so the chromosomes are replicated. So now we have doubles, and they're going to pull apart during meiosis 1. But before that happens, during prophase 1 of meiosis, crossing over happens. So remember, a minute ago, we only had wild type, and mutant chromosomes. There were no wild type, mutant chromosomes at all. But now after crossing over, we can see that we have right here, this chromosome right here is wild type mutant. That's a new combination of traits. Genetic variation has just occurred. Now we can have offspring that look different than our parents. Okay, same thing down here. Instead of having all mutant chromosomes, we have a completely mutant chromosome, but we also have a mutant chromosome with wild type genes on it. So down here, this is the very outcome. This is the same gamete that the parent had. This is the other gamete that the parent had that it got from its parents. Down here are recombinant chromosomes that were not found in the parent generation. This right here is genetic variation. This is what keeps evolution happening and populations changing. It's very key. Here's it bigger. Okay, so here were the 4 eggs that that female could make. Here's the 2 recombinant chromosomes. This guy, the father, is only making one kind of sperm because he's homozygous. So recombination would give him exactly the same thing. But when we do the cross, so we cross our female eggs with our dad's sperm, we see these are the parental types. We're still seeing them. We haven't destroyed them, right? We still have wild type, crossed with mutant, which will give you wild type. Or down here we have mutant, with mutant, will give you mutant. Those are our parental type offspring. That's what we would expect to see, right? And over here is our recombinant offspring. So right here, this is going to be, we have 2 mutant wings. So this guy's going to have short wings, but he's going to be gray. And we did not see any gray with short wings in the previous generation. So this is a recombinant offspring. This is genetic variation. This was not found in the prior generation. Same thing right here, except we're going to have a black body because he has 2 copies of the mutant allele, but he's going to have normal wings because he has a wild type normal wing allele. So to determine, once we determine this, you count the offspring and you see how many are different than the parents, okay, for a linked trait. And we're going to come up with the recombination frequency. So out of all of our organisms, there were 2,300 total offspring. 965 of them plus 944 of them were parental. But 206 plus 185 were new combinations of traits. We had 391 recombinant offspring. So we just figure out what percentage that is out of all of them, what percentage 391 is out of 2,300, and it's 17, I know you can't see, 17% recombination. So that's important. We're going to come back to that. So it says recombinant chromosomes bring alleles together, new combinations of gametes. We were just talking about that, having different gametes than the ones that your parents had. And then random fertilization. Again, this wasn't random because they were doing it in a lab and picking who was mating with each other, but random fertilization does exactly the same thing. Increases the genetic variation by bringing together new traits that were not there before. And it's important to note that if there weren't already genetic variation in the world, then this wouldn't matter, right? If every single fruit fly had exactly the same traits, then we would never get new traits anywhere. So that's why mutation is important, and we're going to get to that in a later chapter. Okay. I'm going to pause. Next video.